Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Laura from Technology Networks, and today we're joined by Dr. Erin Zhang, Deputy Director of the Product and Marketing Center at NGI. Today we'll explore how a revolutionary next-generation high-throughput gene sequencer is transforming the way we understand and approach clinical research, population genomics, and large-scale sequencing projects. From groundbreaking discoveries to real-world applications, we'll dive into the ideas and innovations driving the future of science. So, without any further ado, let's get into the science. Hi Erin, thanks so much for joining us today. So, can you start with introducing the technology? What's the DNB T7 Plus and why is it such a big step forward for genomic sequencing?
Yeah, well, thanks for having me here. So T7 Plus is our next generation powerhouse sequencer. So internally, we call it data mining machine. I would say it would change how labs handle large scale genomics. With T7 Plus, you don't have to choose between scale and speed. It can deliver more than 14 terabases of data in one run while maintaining 24-hour turnaround time. And with an integrated hardware design and a smartphone-like control center in the software, T7 Plus really reduce the technical barriers for the researchers. And this will allow more labs to access population scale sequencing. MGI also provides multi-omics solutions, such as spatial omics, single cell, proteomics, partner with third-party company, methylations and all that. And T7 Plus enables all these solutions in one single sequence run. So this integration is really reshaping how we generate and analyze data, and whereby the concurrent data streams are now feasible for large cohorts.
Wow, that sounds amazing. So, with that in mind, what would you say makes the DNB Seq T7 Plus stand out on the market?
First of all, I would say it's the speed. Currently in the market, there are few high throughput sequences that can generate tens of terabases of data in one run, but none of them can complete within 24 hours. And second, I would say that we completely rethought about the user experience. We not only conducted hundreds of in-depth conversations with our partner clients, we also include ergonomics, user behavior pattern analysis, and even user sentiment analysis to assist the design of instruments, consumable, and user interface. And last but not least, we know that running cost is very crucial to our users. So during the entire R&D process, we place strong emphasis on the cost optimization. So as a result, our users can not only process large number of data in minimal time, but can also benefit from significant reduction of the cost.
Wow, it sounds like it's got some really impressive features. So I'm thinking about its applications and wondering how will the T7 Plus change the game for genomics research?
As I mentioned, that users traditionally have to choose between scale and speed. So they either get quick results with fewer samples, or they handle more samples but have to wait longer time. So this compromise has been slowing down research for years. T7 Plus, I would say, can give you everything, can change everything by giving you both at the same time. Now researchers can process large batches of samples really fast. And the bonus is we make it really cost effective and smarter. So at the same amount of time and budget, researchers now can generate more data. And this means faster discoveries and bigger breakthroughs.
Wow, so you've mentioned it's incredibly fast. What's the science or innovation behind that short turnaround time?
Oh, well, we really rebuilt the sequencing from the ground up. For example, let's say for the flow cell, we designed an ultra-dense flow cell with a triangular array, and this maximizes the number of DNBs on the flow cell. DNB stands for DNA nanoball, which is our core technology. And basically, it means the number of libraries. So this allows significantly boost of the throughput without compromising the imaging speed. And in order to identify the base pairs on the DNBs that are packed so close to each other, we developed a high resolution and high speed TDI camera. And paired with the high performance sliding platform, it can accelerate just the imaging process by 20%. And for the fluidics in the machine, the high speed DNB preparation by our proprietary rig technology combined with smart sensors, it can adjust the reagent flow rate to achieve its peak efficiency under varying conditions. And for data generation, a multidimensional optimized base calling algorithm fully utilizes server capacity and make the data file generation faster by 4 times. So by integrating all these advancements, T7 Plus can deliver the data in such a short time, which other platforms at the same throughput level cannot.
So you've mentioned the word intuitive. How does the T7 Plus help make sequencing more intuitive and accessible for users?
During R&D, I would say we really work from the ground up. And first, we analyze the entire process. So we basically simplify the process with our 7-in-1 design machine. It will include the steps from DNB making, loading, sequencing, all the way to compressed data generation. So this means no more manual DNB prep steps. You get all the benefits from DNB technology, such as high accuracy, low duplication, low index hopping, without extra hassle. And for the reagent cartridge, our modular design minimizes manual pipetting steps. And coupled with RFID auto identification, it will ensure error-free setup for different applications. And most importantly, for the software, we basically ask ourselves the question that what if sequencing is as easy as using a smartphone? So as you can see that our smart control center works just like your smartphone, showing you everything you need at a glance. Even for the first-time users, they can pick up very easily. So by combining all these smart hardware and software features, we've made professional grid sequencing truly easy to use.
Amazing. And who would you say needs to know about the DNB-seq T7 Plus the most?
Oh, well, T7 Plus is purpose-built for high-throughput sequencing scenarios. It can deliver industry-leading speed and throughput. So this makes it ideal for large-scale research projects, such as national population studies, multiomics projects, and it is also ideal for core facility labs at research institutes, commercial sequencing providers, or I would say any centralized sequencing facilities. For clinical scenarios, it can be used for cancer-related tests that requires large amount of sequencing data, such as early cancer screening and post-treatment monitoring. And of course, it can also be used for other specialized fields, such as agriculture genomics and environmental studies.
Great. And then thinking about MGI's previous releases, how does the T7 Plus differ from the original DNB-seq T7? Is it an upgrade or a completely new innovation in genome sequencing?
Well, the original T7 was great. It's still faster than other sequencers at its throughput level, but we weren't satisfied. For years, we kept asking ourselves, how can we make it even faster? How can we make it simpler? And I would say more crucially, we asked the question, how can we better serve our users' real needs? So T7 Plus is our answer to this. And that plus in the name means we kept everything users love about T7 while adding breakthrough improvements, giving users both cutting edge performance and reliabilities.
Amazing. And how does the T7 Plus fit into MGI's existing ecosystem of sequences, and what gap does it fill?
MGI has the most complete lineup of sequences in the industry. We offer both short read platforms and long read sequencing platforms under our seek-all approach. So this means we've got solutions for every genomic sequencing applications you can think of. In this family of products, I would say T7 Plus is our top of the line short read workhorse. It solves a huge problem in the market, giving labs massive sequencing powers that's actually easy to use.
Right, and you mentioned both short and long read technologies. What kind of research or clinical questions become easier to answer when using both T7 Plus and long read sequencing together?
Our T7 Plus, the short read platform, and Cyclone-seq G400ER, the long read sequencing platform, are the perfect pairing. They cover each other's blind spots to give you a complete picture of the genome. T7 Plus brings super accurate and affordable large-scale sequencing, while G400ER delivers super long reads. So together, they are able to unlock the capabilities such as perfect genome assemblies and the ability to crack through different difficult regions on the genome. So if we put it into scenarios, I would say it would be like a better understanding on our genome during large population scale studies and a comprehensive cancer profiling. So small mutations, fusions, CNVs can all be detected in one run. And for rare disease, I would say we can give a better diagnosis even for the tricky regions.
Sounds amazing. That's about all we have time for. Thank you so much for walking us through the new DNB-seq T7 Plus. It sounds like a real advance in genome sequencing technology.
No problem. Thank you for having me here.
Thank you. Thank you to Erin for joining us today. To learn more about real-time multiomics and the full spectrum of digital equipment and systems, please click the link in the description. Thank you for watching. Together, we are shaping science.